If you want to know, like, what's actually keeping a deadly tumor alive, looking at its mutated DNA might actually be the worst place to start.
Yeah, which is, I mean, that's a really tough pill to swallow, considering we've spent the better part of the last few decades doing exactly that.
Right. It completely flips the script. But to understand why, I want you to imagine a towering, highly unstable Jenga tower. It's swaying, it's patched together with all these mismatched pieces, but somehow it is still standing.
Barely standing sometimes, but yeah.
Exactly. Now, if your goal is to bring that tower down, you don't just, randomly flick pieces off the top, and you definitely don't just stare at the broken blocks on the table. You want to find that one specific load-bearing block at the very bottom.
An anchor piece.
Right. The one piece that, if you pull it, makes the entire structure just collapse instantly.
Because the tumor, it might be structurally compromised, but it's highly resilient. It's found all these biological workarounds.
Welcome to the Deep Dive. Today our mission is to map those hidden workarounds, the absolute Achilles heels of cancer. And our guide for this journey is a landmark 2017 study from the journal Cell titled Defining a Cancer Dependency Map.
It's an incredibly massive undertaking. It was led by Aviad Cherniak and a really huge team at the Broad Institute.
Right, and it's a phenomenal piece of work because for a long time, oncology has been so focused on cataloging the broken parts of the cancer machine. You get a sequencing report, lists your genetic mutations, and we try to target them.
But knowing what's mutated in the blueprint doesn't automatically tell you what the physical tumor is actively relying on to survive right in that moment.
Okay, so let's unpack this. A mutation is essentially just a broken window, but we need to find the load-bearing walls. But how do we actually find those critical Jenga blocks across hundreds of completely different types of cancer? I mean, it sounds like looking for a needle in 1,000 haystacks.
Well, you do it by systematically turning off the genes inside the cancer cells, literally one by one, and just watching to see which specific gene deletions cause the cells to die.
Just one by one?
Yeah, one by one. In this study, the researchers took 501 different cancer cell lines, and we're talking about a vast array of diseases here, leukemia, lung cancer, melanoma, pancreatic cancer.
That is a staggering scale.
It is. And they used a technology called RNA interference, or RNAi, to silence these genes across the board.
So the scale of this interference, they used a library of 100,000 different short hairpin RNAs, right? We'll just call them shRNAs.
Yeah, shRNAs is much easier to say.
Right. And I like to think of an shRNA as like a tiny, highly targeted molecular assassin. Its entire job is to intercept the specific gene's instructions before the cell can even carry them out.
That's a great way to picture it. But the physical mechanics of how they deploy these assassins is what makes this data so reliable.
Yeah.
They used a technique involving a low MOI, which stands for low multiplicity of infection.
Okay, wait. Which means what exactly? Like in terms of getting the shRNA into the actual cell?
So they deliver these shRNAs into the cancer cells using viruses.
Yep.
But if you just, dump a whole bunch of virus onto a Petri dish, one single cell might take in 5 different shRNAs.
Oh, I see.
Right. So if that cell dies, you have absolutely no idea which of the 5 assassins actually killed it. So they diluted the viral delivery system to an incredibly precise degree.
To make sure they didn't double up.
Exactly. They ensured that each individual cancer cell received exactly one and only one shRNA.
So it's a perfect 1-to-1 ratio. One cell gets one targeted gene turned off.
Yes. And once that was set, they let these 501 cell lines just grow and multiply for 16 doublings, which took up to 40 days.
A 40-day gauntlet.
A 40-day gauntlet. And after that, they sequenced the DNA of the surviving cells. The logic is just beautifully simple. If a specific shRNA is completely missing from the final population of cells,
it means the cell carrying it didn't make it.
Exactly. It didn't survive. Therefore, whatever gene that shRNA turned off must have been absolutely essential for that specific cancer's survival.
Okay, I have to push back here, though, because the logic sounds really elegant on paper, but isn't RNA interference notoriously, messy in practice?
Oh, incredibly messy.
Right. You send in this tiny shRNA assassin, but it doesn't always hit just the intended target. It can wreak havoc elsewhere.
You are hitting on the exact biological roadblock that plagued this entire field for years. We call it off-target effects. And the root of this problem comes down to a structural flaw known as the seed effect.
The seed effect?
Yeah. Over 99.3% of the shRNAs in this massive library share a tiny 7-letter genetic sequence, a 7-mer seed, with naturally occurring microRNAs that already exist in our bodies.
Okay, I picture it like having a master key cut at a hardware store. You cut a key because you want it to open one very specific lock, which is your target gene.
Right.
But because of the way the jagged edges of the key are shaped, the seed sequence, it accidentally fits into 12 other locks in the exact same building.
Yeah, that's exactly what happens.
So you end up opening all these doors you never meant to touch, causing massive collateral damage to the cell's internal machinery.
Which creates a huge data problem.
Yeah.
I mean, if a cancer cell dies in your petri dish, how do you know if it died because you successfully knocked out the true load-bearing gene? Or if it died because your shRNA accidentally opened 12 other doors and caused toxic collateral damage?
Historically, you just didn't know.
You didn't.
Yeah.
That collateral damage is just noise hiding the real signal. But this team, they didn't just accept the noise. They built an algorithmic tool to filter it out. It's a computational model they named DEMETER.
DEMETER. Okay, so how does it actually work?
So DEMETER is fascinating because it uses a complex mathematical framework to untangle the on-target gene knockdown effect from the off-target seed effects.
Okay.
The researchers intentionally designed their libraries so they had multiple different shRNAs targeting the same gene and multiple shRNAs sharing the same 7-letter seed sequence. So DEMETER could cross-reference all of these overlapping data points simultaneously.
Oh, wow. It's like listening to a live recording of a symphony that was ruined by someone coughing loudly in the audience.
Okay, I like that.
Right. So DEMETER is the ultimate audio filter. It maps the exact acoustic signature of the seed effect, and it perfectly subtracts it from the entire track, leaving only the pure, isolated sound of the specific instrument you actually want to hear.
That's a perfect analogy.
Yeah.
And by applying DEMETER, they effectively rescued the entire experiment. They salvaged the true biological signals from a literal sea of statistical noise.
Okay, so let's do our quick 5-minute mini recap here to lock this in. Bullet 1, we screened 501 cancer lines by knocking down genes one by one using these shRNA assassins. Bullet 2, RNAi has a massive seed off-target problem causing tons of collateral damage. And bullet 3, DEMETER mathematically separated the noise from the true gene dependencies.
Spot on.
So, question for you listening right now, think about your own field, whatever it is. How much seed effect noise is hiding in your data that a smart algorithm could just filter out and kind of changes how you look at messy data?
It really does. Because once DEMETER cleaned the data, the fog lifted. The researchers could suddenly see the map clearly, and they confidently identified 769 strong differential dependencies.
And differential is the key word there, right? We aren't talking about basic survival genes that every cell in the human body needs just to, metabolize energy.
No, not at all. We're talking about vulnerabilities that are specifically required by only a certain subset of those 501 cancer cell lines.
So they're highly specific.
Exactly. And to ensure they weren't just chasing statistical ghosts, they set an incredibly high bar for these dependencies. They used a 6 sigma threshold.
Wait, 6 sigma? Like in a normal distribution, a 6 sigma event is mathematically extreme.
Extremely rare. We are talking about massive screaming vulnerabilities present in these specific tumors.
But I mean, that introduces a completely different, huge logistical problem. Finding 769 highly specific vulnerabilities is great in a lab. But how do we know which tumor has which vulnerability out in the real world?
Right, because you can't run a 40-day RNA interference screen on every single patient who walks into an oncology clinic.
Exactly. You'd need to be able to predict the vulnerability based on something you can easily and quickly measure right now, like a standard tumor biopsy.
And to bridge that gap, the team introduced another computational tool. This one is the Atlantis algorithm.
Atlantis. I'm sensing a very strong mythology theme with this broad institute team.
Yeah, they definitely have a naming convention going on. So Atlantis is a nonlinear regression model, and it's built on something called random forests, or conditional inference trees.
Okay, let's break that down, because random forest sounds like we're wandering into the woods.
Well, think of a single decision tree like a flowchart. If A is true, go left. If B is true, go right.
Right, pretty standard.
But a random forest is an algorithm that generates thousands of these separate decision trees, feeds them data, and then has them all vote on the final prediction.
Oh, so it's a consensus system.
Exactly. They took 66,646 molecular features from these cancer cell lines, We're talking about a massive data set of DNA mutations, the number of copies of specific genes, and the RNA expression levels.
And they just fed all of that into the forest.
All tens of thousands of these features went into Atlantis to see if it could predict those 769 hidden dependencies.
It is essentially playing the world's most complex game of 20 questions with the tumor's molecular profile to guess its hidden weakness. And it worked, right? The paper says it successfully modeled 426 of them.
It did. But the actual features that Atlantis chose to make those predictions, that is what really shook the field.
Yes. Here's where it gets really interesting. Because in oncology, as we mentioned earlier, we have been obsessed with DNA.
Obsessed.
We assume DNA mutations are the ultimate undisputed drivers of everything in cancer. But when Atlantis spit out the absolute best predictors for these vulnerabilities, DNA mutations were only the top biomarker 16% of the time.
Just 6–10%.
And copy number variations, meaning how many physical copies of a gene the tumor has, were the top predictor only 2% of the time.
What's totally fascinating here is the overwhelming winner. At 82%, the top predictor was RNA expression.
82%?
Yeah. The sheer volume of RNA being produced by the cell was the ultimate indicator of what the cancer relied on to survive.
That feels like a massive paradigm shift. Why is that so shocking to the field? But then when you think about it, it makes complete biological sense.
Well, we connect this to the bigger picture. Think of DNA as the architectural blueprint sitting in the construction site office.
Okay.
A mutation just means there's a typo written on the blueprint. But RNA expression, that is the actual concrete being poured. It's the steel beams being actively welded. RNA represents the active state of the cell.
Oh, that makes so much sense.
Right. It tells you what the tumor is physically doing right now, regardless of what the original blueprint in the office said.
Yeah, the blueprint might say, build a door, but if the RNA isn't actively bringing wood and hinges to the site, there is no door. So if I'm tracking this, Atlantis proved that the active work order, the RNA, is a vastly superior predictor of a tumor's weakness than the static DNA blueprint.
Vastly superior.
Which really forces you to reevaluate how we currently prioritize DNA sequencing as the sort of holy grail of tumor testing.
It demands a complete reevaluation of our clinical diagnostics, honestly.
Okay, let's lock in our second 5-minute mini recap. Bullet 1, 769 distinct, extreme, Six Sigma cancer vulnerabilities were found. Bullet 2, Atlantis acted like a massive game of 20 questions using random forests to predict these vulnerabilities using over 66,000 features. And bullet 3, the big one, RNA expression, not DNA mutation, was the ultimate predictor 82% of the time. So my question for the listener, does it surprise you that the blueprint is less predictive than the actual work order? What does this mean for how we currently test tumors?
It's a question every oncologist needs to be asking right now. Because once Atlantis could reliably predict these vulnerabilities, the researchers zoomed out and noticed a larger pattern in the data.
The 4 classes.
Exactly. These hundreds of distinct weaknesses didn't just exist randomly. They naturally grouped themselves into 4 distinct buckets or classes of vulnerabilities.
Right, and this is where we really start seeing the underlying logic of the cancer machine. Let's walk through these 4 classes because the biological mechanics of each are just wild. Class 1 is the one we are actually most familiar with, mutation-driven dependencies.
Right. This is your classic oncogene addiction. These are genes like KRAS or BRAF.
The usual suspect.
The usual suspects. When they mutate, the protein they produce gets structurally locked in the on position. It constantly screams at the cell to divide and grow.
But because that signal is so overpowering, the cancer rewires its entire survival infrastructure to depend on it, right?
Sure.
It becomes totally addicted to that continuous signal.
Exactly. And a great detail from the data here was the discovery involving the GNAI2 gene in diffuse large B-cell lymphoma.
Oh, yeah, the single amino acid thing.
Yes. The researchers found that a very specific deletion of a single amino acid, a mutation known as PK272-del, alters the protein's shape just enough to lock it into that active state.
Just one missing amino acid?
One missing piece. And it creates a total addiction to that exact gene. If you shut it off, the cell just collapses. Yeah. That is a perfect class 1 target.
But class 2 is where the cancer's own biology starts getting really clever. Class 2 dependencies are called CYCLOPS genes.
This is one of the most brilliant biological phenomena we've ever discovered. CYCLOPS vulnerabilities aren't about the tumor trying to turn something on. They are about the collateral damage of the tumor trying to turn something off.
The visual memory here is perfect. I want you to think of the mythological Cyclops, the giant monster with only one eye. He is massive, he's terrifying, but he has a glaring single point of failure. Poke out that one eye, And he's completely blind and defeated.
And the cellular mechanism driving this is based on something called homozygous loss.
Homozygous loss. Okay, explain that.
So in our genome, genes live in neighborhoods next to each other. Often, a developing cancer cell desperately wants to delete a tumor suppressor gene, which is essentially to brake on cell division.
It wants to cut the brakes.
Right. But cancer mutation is a sloppy, chaotic process. When it deletes the tumor suppressor, it accidentally deletes one copy of an essential survival gene that just happened to be sitting right next door.
It demolishes the bad neighbor's house, but swings the wrecking ball way too wide and takes out half of the good neighbor's house in the process?
That's it exactly. Normally, a healthy cell has two copies of this survival gene. Now, the tumor has successfully removed the brakes, but it only has one copy of the survival gene left. It's become a cyclox.
It only has one eye left.
Yes. It is surviving, but it is entirely dependent on that single remaining copy. If we design a drug to inhibit that remaining copy, the cancer cell dies immediately because it has zero backup.
But wait, what about the healthy cells?
The normal healthy cells in the patient's body survive the drug perfectly fine because they still have their original two copies. The paper highlights genes like PLR2A and PSMC2 as prime CYCLOPS targets.
Man, it is literally weaponizing the tumor's own sloppy collateral damage against it. I absolutely love that. Okay, moving to class 3. Expression-driven dependencies. The paper also calls this lineage addiction.
And this loops directly back to our finding about RNA expression being the ultimate predictor. Tumors frequently become addicted to master transcription factors.
Which are what, exactly?
They are the fundamental control switches that define their original tissue type when they were first developing in the embryo.
So the tumor essentially remembers where it came from and clings to those specific identity switches for dear life.
Exactly. The paper notes the FOXA1 gene in breast cancer, or the SOX10 gene in melanoma.
And the strategic advantage for drug developers here is the concept of targeting dispensable organs, right?
Yes. We can safely design a drug to attack the FOXA1 dependency in breast cancer, or SOX10 in melanoma, because a human adult can actually survive without those specific normal tissues functioning perfectly.
We can endure the side effects?
Look, we can. But the cancer cannot survive without them at all. Its entire Jenga tower is resting on that tissue-specific identity block.
Which brings us to evolutionary backups. Class 4, paralog deficiency.
A paralog is essentially a genetic twin. Over millions of years of evolution to protect us from environmental stress, some of our genes duplicated. This gave us two different genes that perform the exact same job. It's built-in biological redundancy.
I think of it like a twin-engine airplane. If one engine naturally fails mid-flight, the plane doesn't crash. It just relies 100% on the second engine. The passengers might not even notice the shift.
Right. And in cancer, sometimes one of these paralog genes, one of the engines, is naturally silenced or deleted as the tumor mutates and evolves. The tumors are completely fine. It keeps dividing and flying along on the backup engine.
But this reveals a massive hidden vulnerability.
Huge. We can design a drug to specifically knock out that remaining backup engine. The tumor crashes and burns. But the healthy cells in the patient's body, which still have both engines functioning perfectly, they just switch to their primary engine and survive the drug unharmed.
Okay, time for our third mini recap. Bullet 1. The 4 classes are mutation-driven, CYCLOPS, expression-driven, and paralog deficiency. Bullet 2. CYCLPS targets the collateral damage of tumor gene loss. And bullet 3. Paralog deficiency exploits lost backup systems.
Perfect summary.
So audience question, which of these 4 vulnerabilities do you think offers the safest path to a drug with the fewest side effects for the patient? It really frames the future of the entire industry.
It does. But it also raises an important question about how we actually move from computational models in a lab to real-world biology in a patient.
Right. Does it actually work in living cells?
To prove this map wasn't just abstract mathematical theory, the researchers dove incredibly deep into a specific clinical case found in their data. It was a paralog deficiency involving 2 twin genes called UBB and UBC.
Okay, let's look at the mechanics of this case study because it perfectly illustrates the whole twin engine concept. Both of these genes, UBB and UBC, they do the exact same job. They produce a protein called ubiquitin.
Right, which is absolutely essential for cellular trash disposal.
Right. Its job is to tag damaged, misfolded proteins so the cell knows to grind them up and recycle them. If you stop the trash disposal system, the cell literally chokes on its own toxic waste and dies.
So we have our twin engines, UBB and UBC. The researchers zeroed in on ovarian cancers and found a fatal hidden flaw. In many of these specific tumors, the UBB gene's promoter was hypermethylated.
Okay, let's define that. Methylation is an epigenetic change. I picture it like slapping a heavy chemical padlock onto the DNA switch.
That's a great way to describe it.
It doesn't actually mutate or break the DNA sequence itself, right? It just permanently locks the gene in the off position so RNA can't read it.
Exactly. And because the UBB engine is padlocked shut by this hypermethylation, the ovarian cancer cell is flying entirely on the UBC engine. It is relying 100% on UBC for its trash disposal.
And here is the physical proof they found, which I find just amazing. When the researchers experimentally knocked down UBC in these specific low UBB ovarian cancer cells, the total levels of conjugated ubiquitin, which simply means the ubiquitin tags that are actively attached to the trash, plummeted.
Yep. The trash immediately piled up.
And the cancer cells died rapidly. But, and this is the real smoking gun, if they artificially added the UBB protein back into the cells, essentially unlocking that first engine.
The cells easily survive the UBC knockdown. It perfectly validates the Atlantis algorithm's prediction.
Wow.
The hypermethylation of the UBB gene is the exact measurable biomarker that tells a clinician, hey, this specific tumor is uniquely vulnerable to a UBC inhibitor drug.
So what does this all mean for the future? I mean, the paper successfully mapped 769 vulnerabilities. They proved it works with UBB and UBC. Is the map done? Can we just print the textbook and start manufacturing the drugs?
Oh, not even close. In fact, to figure out the scale of what a complete future map would look like, they ran a downsampling analysis. They proved that 501 cell lines, as massive as that sounds, is literally just the starting line.
Yeah, they looked at the statistical curve of discovery, didn't they? You see an inflection point around 200 to 300 cell lines where you start making real, recurring discoveries.
Right, but human biology is incredibly diverse. To simply observe all the major dependencies that exist across the vast spectrum of human cancers, the math dictates we need to screen at least 1,000 different cell lines. And observing them is only half the battle. To have the statistical power to predict them accurately with molecular features, to train the Atlantis algorithm properly so it can be used flawlessly in a clinic, the data indicates we need a massive international effort to screen over 5,000 distinct cancer cell lines.
That is it is the ultimate biological cartography project.
Truly.
Okay, final 5-minute mini recap. Bullet 1. UBB and UBC are redundant ubiquitin genes. Bullet 2. Ovarian cancers frequently silence UBD via methylation, making them 100% reliant on UBC. And bullet 3. We need an international effort of over 5,000 cell lines to actually finish this map.
Nailed it.
So, listener, if you were designing a drug today, would you target the tumor's genetic mutations or its paralog deficiencies like UBC? It's a huge shift in perspective.
It's a daunting scale, but it's a question that is currently reshaping pharmaceutical pipelines worldwide. Are we going to keep chasing messy genetic mutations? Or are we going to build drugs targeting these structural dependencies where the cancer has already lost its backup engines?
It really is a shift in philosophy. To wrap up today's deep dive, what we've seen from Cherniak and the Broad Institute team is how computational algorithms can take incredibly noisy, messy biological data, strip away the interference, and hand us a precise map of cancer's Jenga block.
We aren't just blindly guessing which blocks to pull anymore.
No, we are calculating the structural fail points before we even touch the tower.
Exactly. And if we connect this to the bigger picture, I want to leave you with a final thought to mull over. If this dependency map is eventually completed across those 5,000 cell lines, it will fundamentally change our taxonomy of disease.
How so?
We will likely stop categorizing cancers by where they happen to live in the body, like lung cancer or breast cancer. Instead, we'll categorize them by their algorithmic dependencies. A patient won't be diagnosed with ovarian cancer. They will be diagnosed with a paralog-deficient, UBC-dependent tumor, or a vulnerable tumor.
Oh, wow.
Right. If cancer is ultimately just a complex machine with predictable mathematical fail points, are we finally looking at the actual blueprint to safely dismantle it?
And suddenly that wobbling, terrifying Jenga tower doesn't look quite so intimidating anymore. You just need to know exactly which block to pull. Thank you for joining us on this deep dive. Keep questioning your assumptions about how we fight complex diseases, and we will see you next time.